we've actually kind of put together an idea of how this could be implemented. Now, I don't want you to feel that we've chipped this into granite. This is not hardwired or hard-written. This is not the 10 commandments of laser microdissection that everyone from now on out will have to follow. This is just an idea. It's a goal. Can we put all these different things we've learned, like the fluorescence and the cutting and the sample, the extraction, all this into a comprehensive process? One, the main point is to improve processing of sexual assault evidence using laser microdissection microscopy. Can we expedite the processing? Kelly says yes. He said, okay, we can leave out all these different steps in it, but we're still getting good separation. Expedite the processing. Can we enhance the capabilities? Well, I talked about the fluorescent sperm identification. Well, okay, if we don't find sperm, can we now look at it with a little bit better technique for finding sperm? Enhance the process using for difficult samples. And in doing so, in this goal, can we have a comprehensive process that also takes advantage of new capabilities, the ability to take the diploid cells and separate them? Is this something that can be done? Can this be done on things like vasectomized males or sexual assault where you have vasectomized or azoospermic males, where you can maybe perhaps find the white blood cells or epithelial cells in the male in the vaginal samples? I mean, we can't do that now. We can do YSTRs, but if you've done them, if you've known the wonderful statistics they have compared to getting a random match probability. So we've looked at this and discussed it now for a couple of years, and we have kind of an outline or the basis of a process of how to do all of this and how to put it all into one comprehensive process where you can take advantage of it and you can make decisions based upon what you learn at each step. You can start this at this process, but you don't have to finish it. You can go other ways. You can make decisions along the way, but they're educated decisions. They're decisions based upon more knowledge of the sample that you have. Here we look at the overall outline of the project, or kind of a process to kind of talk about why and where I'm coming from. Here you have vaginal swabs, washings, breast, bite, neck, breast swabs, oral, rectal swabs, suspected semen stains, urogenital stain swabs, and fingernail samples. Okay, these are the different types of samples that you normally get during sexual assaults. The neat thing about this in sexual assaults, almost always have the potential for mixed DNA. And fortunately, in most sexual assaults, it's going to be male and female mixed DNA. So that's one of the things here. We have sexual assaults. They almost always have some type of mixed DNA, and almost always it's a mixture of male and female DNA. And it's not 100%, but most of the time that's what it is. If we look at the type of analysis that we do on them now, or what type of cell mixtures we have, we take the vaginal swabs, washings. Typically, we're going to be looking for something like a sperm epithelial mixture, so we're going to do a differential extraction on that. Bite, neck, breast swabs, typically that's epithelial mixtures, but they may be semen epithelial mixtures. You don't know that unless you read the history or someone says, you know, that swab I took from the chest is actually, I suspect that to be oral semen mixture. So that would make a difference on how you would extract it, wouldn't it? You wouldn't use a standard straight extraction if you thought there was spermatozoa in there or semen in there. So, okay, but most part, it's going to be a standard or straight extraction, and that's why I had the question mark by there. You're not always certain. The other one, oral rectal swabs, sperm epithelium typically, differential. Suspected semen stains, sperm epithelium, epithelium, again, possibility mixture of using a differential there. Urogenital stain swabs, typically you would suspect those would be a sperm epithelium, but they could be epithelial mixture. Again, differential typically. Fingernail swabs, those are typically going to be an epithelial or epithelial blood type sample where you could do the standard extraction. But if they're mixed stains, you're not going to be able to do anything with that. In other words, you're not going to be able to resolve the mixture except for hope that the suspect is the major contributor. So we look at it, differential extraction. Most of you have worked with differential extractions used for semen-containing samples. Hopefully the sperm in there, because it's really used to separate the sperm from the other cells. The strengths are that it separates sperm and epithelial DNA. It also contains components for the microscopic identification of the cells. You can look at the microscope and you can see spermatozoa and or epithelial cells. It has the internal QC check that if you do test the epithelial fraction, you can confirm that that went back to the, goes back to the victim. Weaknesses, cellular extraction may not get all of the cells off the specimen. We find on cotton swabs you're only getting about 25 to 30% of the cells off the cotton swab if you're doing a cellular extraction. So you're not getting all the cells off the swab to begin with. Consumes part of the sample for microscopy and quantification. Use of, at least as you spot it now, a lot of people will spot 10% of their sperm pellet just to examine for sperm. It's user intensive and complex. There does take a little bit of user training in here. When you're doing those sperm washing, you may lose some of the sample. Almost always when you're doing this procedure, you have to be sitting there doing it. There's not have a lot of dead time or time for something else. Like Kelly was talking about, you run the procedure and you walk away, come back 3 hours later or the next day. Cannot always get a clean separation. Whenever you have very few spermatozoa, often it's very difficult to get a sample that's entirely free of epithelial DNA. You can try, but often it's much more difficult. Or sometimes it's virtually impossible. Cannot separate non-sperm cell mixtures. So if you had an instance of a vasectomy, you're not going to be able to get a random match profile of a clean separation unless you're really, really lucky and happen to the examiner happens to take a almost entire semen sample, pure semen sample during the hospital examination. So if you have those types of samples, you cannot get a good clean separation or separate those types of samples. Again, the novel process that we're talking about, all these steps are possible. The standard extraction used for non-sperm containing samples, the strengths, you're going to get just about all the DNA off of this sample. You can digest sample directly with good DNA recovery. In other words, you get a really good recovery by digesting the whole swab instead of trying to do a cellular extraction ahead of time. But what you lose in that, you don't have the microscopic part of it. It's a very simple procedure. You just put your sample in the digest buffer and let it sit for several hours to overnight and then go through the purification process. Not a whole lot of hands-on work there. It's very simple and quick. Weaknesses, cellular extraction is not often done to identify cells. So if you look at a sample and you put it in a straight extraction when it really had semen in there or sperm in there, now you've got a mixed sample and you can't go back and start doing the differential extraction with that. Cannot identify mixtures prior to data analysis. If you look at a blood stain or something like that, or a stain on a person's clothing, and you run the sample, all of a sudden, oh, it's a mixture. Nothing, there's nothing you can do about that. Unable to resolve some mixtures. Some mixtures are so complex that you really can't resolve them. Even ones that maybe only have a very low level of minor contributor, some way to enrich the minor contributor, that would certainly help. And presumptive testing may require resampling. Some people or some agencies will actually cut out a piece of material and do presumptive testing on that. Based upon that presumptive testing, then they go back and cut out another sample and actually do the DNA analysis on that. So again, you may have to do, in some cases it could cut out this presumptive testing and then resampling. If we look at the laser microdissection process, the one one that Kelly talked about, we start with a cellular extraction, and one of the differences here that you may not see on the differential extraction, we're going to spot 100% of the pellet. The whole pellet's going on the slide. Typically now you may spot from 5 to 10% of your pellet just to examine for sperm. Some people do, some people don't. But in this case, we're going to put the whole sample on the pellet. We're going to take the whole pellet, put it on the slide, and then we examine it under the microscope where if we find the cells that we want, or put it on the slide, then we can do whatever staining we need to. If we find cells that we want to use, then we're going to do cellular dissection, and then go into a pre-amplification digestion, and then go directly to amplification. You're leaving out epithelial digestion, purification, quantification on there. So that, in that sense, has streamlined this process quite a bit. The laser microdissection, what do we feel on this? Well, all the samples, all types of sexual assault samples are possible. You can take a bite mark breast swab, do a cellular extraction, put it on the slide, examine it. If you don't see any spermatozoa, you can then go directly to some type of diploid cell staining. Strength, clean separation of sperm and epithelial cells. If you do see the separation, you can go ahead and do the dissection. If you don't, then you can decide if you want to do other types of staining, or you may simply decide you're going to cut out the membrane and go directly to a standard digestion. You don't necessarily have to go to the fish if you don't want to or you don't feel like you need to. Or you could go to the fish if you wanted to get a good clean separation. And you thought there would be, you thought there was a bad potential of a really good mixture or a really bad mixture there, you could look at it that way. Microscopic identification of cells increases sensitivity, minimizes handling. You're not having to go from tube to tube to tube to tube, switching things out, doing cleanups, doing purifications, going to easy one tube, or doing phenol where you're going to multiple tubes, going into butanol or centricons or things like that. You basically can do a one-step extraction after cutting out some cells. Quantification is done by cell counting. Fluorescent attachment provides new capabilities. Again, if you want to go to the diploid cell, the fish, diploid cell fish, you could do that. In certain samples, it certainly would be helpful. Weaknesses, it requires cellular extraction to prepare the slide. As I told you, in standard extraction, you can just simply put the sample in the digest buffer and digest it. In this case, with these samples, you're always going to have to do a cellular extraction or get the cells off the medium or substrate. It does require nucleated cell identification, and it cannot separate same-sex epithelial cell mixtures. Well, guess what? You have to do that now anyway. There's nothing novel about that. Even in your other one, even your other testing requires nucleated cells and or spermatozoa. In this case, and you cannot separate same-sex epithelial cell mixtures. Well, you can't do that now anyway. So it may be a weakness, but those are the general weakness of all the extraction processes now. So what happens with this? In bright field microscopy, you can start with that. Faster analysis. You can do the microscopy, cell separation, and purification performed in a single step. You just basically separate your samples by cutting them out, especially the the spermatozoa. Just cut them out, put them in the tube, they're separated. Eliminate DNA quantification. And as Kelly and Christine have shown, when you're able to cut out those spermatozoa, you do get better DNA separation. If you can find 50 spermatozoa to cut out, you'll almost always get a pure, clean DNA profile from the male. The fluorescent microscopy does add enhanced capabilities and new capabilities. Fluorescent labeling of spermatozoa, easier and faster identification. But again, it's not something you have to do in every sample, but the capabilities are there. If you feel you want to do that on a sample, or you could screen, you could make 8 or 10 slides in a day. And then the next day you're going to screen the positive ones. The negative ones, you may say, okay, these 2 negative ones, I'm going to do the sperm paint or sperm highlighter on and examine them tomorrow. But the 8 slides I've already got sperm on, they've already been cut and separated, and they're ready to run on the electrophoresis by the time the next day you're ready to run the other samples. And the fluorescent labeling of X and Y chromosomes. If you have cases like fingernail scrapings or bite mark swabs, you can go directly to that. You don't have to go through these other things, other procedures, but you could do an initial examination in a bright field, and you don't see any evidence of spermatozoa, just go directly to the fish. But the neat thing is, with the laser microdissection and these microscopes, you can combine both phases into a single comprehensive process. In this case, you can make it a sequential step where every time you go down a step, you make an educated decision. This is what I'm going to do based upon what I've seen from the previous step. Here we look at going back to the LMD analytical process. I've kind of condensed it down. We now have the cellular extraction. But you know, we still have the capability of doing presumptive tests because we just do it on the supernatant. We do the cellular extraction in PBS or water. We take the supernatant out. We can do AP, P30 if we want to. After the cellular extraction, we then mount it on the slide. We're going to put the whole pellet on the slide and examine it. Now, instead of doing 5 or 10% of the sample, we can have the potential of looking at 100% of the sample. So maybe in your initial spotting of 10%, you found 3 spermatozoa. Now just think if you're spotting 100%, you'd be up to 30 spermatozoa on average, which may be enough to do the full testing, whereas the 3%, you wouldn't have it. We couldn't do it. The next start, we have the examination, where we microscopically identify the cells. Then we go into our cell dissection. Here we dissect appropriate cells directly in the PCR tubes, and then go into pre-amplification, digestion, amplification. All just a very quick, straightforward procedure. What we really want to concentrate on, though, in the laser microdissection process, in other words, from cell dissection, from this point down, we're pretty much the same. And from this point up, we're pretty much the same. In other words, we're going to cellular extraction, spot 100% of the cells on the pellet, or the pellet on the slide. That's going to be the same no matter what we do with the sample. And once we've cut out the cells that we want to, then we go all the way down to the amplification. So that's pretty much the same. It's this point here where we're going to be able to make our decisions based upon what we see. So now we come over here to microscopy, identify cells. This is the point we're looking at. We have the slide we've prepared. We have the analyst looking in the microscope. Obviously, that's not an LMD microscope, but it's still, you got to get the gist of it, microscope. Okay, the first step, we look at stained or differential interference contrast or phase contrast with new slides, identify epithelia or spermatozoa, and cut them out. At that point, if we've made those identifications and dissected the cells at that point, we can simply stop. We go on to DNA analysis. And the little check mark there for the DNA says, okay, that's been done. That's been verified. We can cut those cells out and we can do the DNA analysis on them. That's been established. We've done that. Christine and Kelly have done that. We know that. That's a known topic there. We can do the work on that, or we can do the analysis. The next step, if we do not see any spermatozoa, but then we can, if we choose, we can do the fluorescent identification step. We can, again, like I said, you start with 10 slides one day, you examine 8 of them are positive for spermatozoa, you've done the cutting, you've isolated the cells, and you can start the amplification process. 2 of them were negative. You couldn't find sperm on them. Take those 2 and do the hybridization process of sperm paint overnight. The next day you can come back through and look at them and say, hey, here's some spermatozoa I missed the first time around. Then you can cut them out and start the process. Now that is one of the processes now. Oh. That process now has not been verified for DNA testing. I have not cut samples out and been able to get and tested them for DNA. We did try it once on the director slides, as I told you, it was unsuccessful. But it's still, I think DNA analysis is going to be possible in these slides or these stains, because basically you're staining it with a protein and nothing else. And I know sperm paint is simply put it in water and put it on the slide and let it go. So I don't think there's anything in there that's going to inhibit DNA processing. But at this point, this is the only step that hasn't had the DNA testing verified on it. If we look at the stain, say we're looking at a bite mark or a breast swab or a fingernail scrapings, we see no spermatozoa. We're pretty confident that we're not going to be spermatozoa. If we want to stain this, we can. We then can choose to go to the chromosome paint. we can look at the slide and see, okay, am I seeing male and female cells? If so, I can cut the male cells into one tube and the female cells into another tube, based upon what I see with the chromosome staining. But again, we do this process, first process, second, and third. All those can be done sequentially without harming any of the previous steps. That's what we hope to see. Again, this is something we're looking at to verify. So what we're hoping is, okay, we do step 1, step 2 if we want to, go to step 3, and if we can identify male and female cells and separate them, do DNA testing on them, we're fine. If all of those procedures fail, the important thing about Kelly's talk is where we cut that membrane off of there. Can we still do the DNA analysis on the slide itself? Yes, we can. And remember the slide I showed you, after we cut the X and Y, after we cut the chromosome paint, after we did chromosome painting on there, we then cut it off, and we were still able to do the YSTR testing. So even though we're using the Y-DNA probes, it doesn't affect the YSTR testing, and it doesn't affect the other DNA testing. So even at this step 4, even if all the other previous steps are negative, and we still want to test it, we can. We simply cut the membrane off, put it in a tube. We could do a differential extraction, or we could do a standard extraction. They both work the same. There's no loss just because we put everything on that slide. That was one of our main concerns, is if we do mount on the slide, are we going to lose anything? No, we don't. But we gain the capabilities of laser microdissection all the way through there. So even if you did nothing else but spotted on the slide, looked for sperm, and then cut it off and did your differential, it would be the same as if you had just taken the stain, spotted some of it, and done the differential out of the tube. So we haven't lost anything. But yet, look at all the things we can gain. We can gain the advantages of cutting it out under bright field microscopy. We have an advantage of gaining the ability to see few spermatozoa in the sperm paint. And then, if necessary, we have the advantage or the new capability of being able to separate male and female cells on there. So, yes, we could go directly from the slide to number 4 without any loss. But if we go 1, 2, 3 in a sequential, comprehensive process, we use all the capabilities and all the advantages gained by laser microdissection. And again, we can always go back to the 4th step and do the standard differential DNA analysis that we've been doing all along. So again, novel process, prepare the slide, examine under bright field optics. If we see spermatozoa, go ahead with laser microdissection. If we don't, then we can use the fluorescent capabilities. We can do fluorescent optics, sperm identification. If we see sperm under the fluorescent optics, under fluorescence, we proceed with laser microdissection and PCR. If not, we could excise the entire smear. In other words, if we had a vaginal sample and we weren't able to find spermatozoa, we could then go back to the differential if we wanted to. Or, again, we can go into the chromosome paint. It's a little bit more involved process, but you may decide on some samples that that's what you want to do. But again, all along the line, you can make informed decisions. Decisions because you have information about what's in the sample. And, you know, if last but not least, you can cut out the membrane and do just the regular extraction you would have done normally. And again, now I'll look at this chart where we had the differential, or we do the, in the first part we had differential or standard, or sometimes we weren't sure. Now, with this process, we can look at the whole picture here, and this microscopy will fit for every sample on there. You just go to the vaginal swab, vaginal washings, sperm epithelium. You can examine it under bright field optics, or I put DIC there. And hey, you find spermatozoa, you're fine. If you don't, you still have the option to do sperm paint and chromosome paint. If you go to the bite mark breast swabs, you still start out the same way. You can do the bright field objectives. Do I have any cells there? If I got a bunch of cells there, do I want to go ahead and do the chromosome paint to try to get good separation? I can jump right to that. If nothing else, when I'm through with that, I find go in there and the stain doesn't work or the cells, I can't identify any, I can still cut it out and do the same standard extraction on it. But I have confirmed that I don't have spermatozoa, so I may not have to use another type of stain. In other words, I don't have to use the differential. Oral rectal swabs, the same thing. Start out with the process. Is there sperm present? Cut them out. Otherwise, you can use other types of staining or other tests. Suspected semen stains, same thing. Urogenital stain swabs. The whole procedure is basically you can use that process and step to any procedure you need. You start with a bright field, look at it. You see all epithelial cells on the fingernail swabs. You then just simply do your staining, do your chromosome paint. You don't have to go in there. You don't have to do any enhanced staining for spermatozoa or anything like that. Just a quick glance. I've got all epithelium. Now I'm going to stain them for the chromosome paint. If we look at the slide, I'm not sure if these numbers correspond to what Kelly had, but prepare slide 1 to 2 hours, examine under bright field microscopy. I have 30 to 60 minutes. Fluorescent optics. For sperm paint, that'd be about 30 minutes plus overnight plus examination or re-examination. Chromosome paint takes about 30 minutes ahead of time, although you do have to have some reagents pre-prepared for the chromosome paint. 30 minutes plus overnight plus about an hour after the next day, and then examination. And pre-amp, PCR, and gel is about another day. So basically, I think, Kelly, would you agree this is starting one day is about the end of the second day you would probably have some kind of results?
Yeah, I think when you add in the chromosome paint or whatever, it might,
that might take you to the third day.
Because that preamp digestion, at least the way I have it right now, is 3.5 hours. The amplification itself is about 3.5 hours. So you could see if you were doing a gel, you'd be working hard that day.
Mm-hmm. So basically, you could have, if you found sperm or what you needed in the bright field, you'd have the results at the end of the second day. If you're doing the chromosome paint, then that's going to maybe add another day because you have some overnight procedures to it, or the sperm paint, the same thing. But again, this is a novel process. It may change. It's kind of like in research. You have a hypothesis. This is how we hypothesize this process now. It doesn't mean that when we start doing it, things won't change. Ideas won't, or new information may change part of the process. But the validation, comprehensive process, we've looked at several things about sample preparation. We do know that you need to do a cellular extraction. We talked about stability on the slide. We've seen some issues with the director slide of how slick they are. Are they stable for long periods of time and dried on a slide? My initial guess would be yes, but it's certainly something that would have to be examined. Validation of individual components. I think Kelly's quite a long way on doing the sperm identification and separation. I know we have a few more months or so to finish that up, but it's certainly a good process now, and we're getting 99% allele recovery or identification on 50 sperm. I think that's the benchmark right now. We have something that we can measure against. When we first started 2 years ago, we didn't have that benchmark, but now we can say 50 sperm, 98% of the time we're going to get allele calls. So if we improve the process or change it, we have a benchmark to measure it against. Are we improving or are we going downhill? The individual components, Jennifer published the part on the fish, on the exobyte cell. We know it's doable. I think it's better, can be done better or cleaner, but it's doable. Now, we may have to validate that individual component. We also have to validate the sperm identification, the chromosome paint, or not sperm paint or sperm highlighter. But I think all of those components are doable, and all of them have at least been shown to be working on evidence material. We've also looked at the between component interferences. If I do does the nuclear fast red interfere with the sperm paint? Does the nuclear fast red interfere with the chromosome paint? So far, we haven't seen any interferences in any of those. In other words, we can still do chromosome paint even after having stained all the nuclei with nuclear fast red. We've seen sperm paint will label those spermatozoa that have been stained with nuclear fast red. We don't know if we do sperm paint and then go to the CEP or the fish, is there any effect there? Will that enhance or inhibit our work with the X and Y chromosome? That's between component validation that we need to look at. And then we look at the whole integrated process. Are we really not losing something? Are we really not losing anything? So that's an issue. And again, is anything lost by losing this process? And can this be validated incrementally? And I believe certainly the bright field part of it is a long way, and that will, as Kelly says, focus on that, get that done, and let the others fall into place. We know that can be done by itself. And we know that that is working quite well right now, and it's just a matter of putting some more effort and time into finishing it up. And again, these are the people that have worked on this and the crime lab. And any questions? Discussion? Otherwise?
I like the process of, I was wondering, you kind of mentioned, Christine, that sometimes it might be useful to dilute. You know, or maybe I thought I heard that. Before smearing an entire sample, just so you could spread out and maybe select more easily.
Right.
And, you know, your process may not accommodate that in its simple view. I mean, had you thought about that, or?
I mean, we've never really, I know Kelly's spotting 100% right now. Have you had any problems with it?
What I'm doing is I'm doing about a 25 microliter spot. I haven't actually gone to spotting like an entire, pellet. But of course, a lot of times with vaginal swabs, maybe that we can just put a procedure in place, but most of the time your vaginal swab is going to have a lot of cells. Maybe we could just put a procedure in place where instead of taking off all your supernatant except for, I think most people do all but, let's say, 30 to 50 microliters, maybe it'll be all but like 100 or so. Because always if, you could, or at least spot partially, do a 3 or 5 microliter spot and look at it and then spot the rest or something. Right. Yeah, we're going to have to play around with that definitely to see if maybe a more dilute pellet would be better.
There are other issues. I know when Jennifer was doing the fingernail swabbings, we swab on the fingernails, that they were getting more clumps of cells, and we were having to make an effort to break them up. Well, what we were doing to break them up was running through a cell filter, a kind of filter used before flow cytometry to break apart the clumps of cells. But I've also seen issues, and I have documented issues in some of the work we've done, is that those also capture spermatozoa. So, there are some sample preparation issues beyond just shaking it up and putting it in there. But also, like your bite mark breast swabs, those often have very few cells in them. So we certainly wouldn't have to worry about diluting those. But you're right, we've had some vaginal samples. I know Kelly has really started liking the vaginal washings. Our rape kits get vaginal washings.
I was going to say, in Louisiana, we actually still get vaginal washings. And actually, after I started doing LMD, I actually encouraged them to keep it because, gosh, that's a lot easier than having to take this, the sample off the swab. We can just take the washing now, put it, pellet it, and that's it, I mean, that's kind of nice for LMD. Yeah, I don't know if, I'm getting funny faces. Does anyone know what a vaginal washing is? I didn't know what it was until I went to Louisiana.
She not only encouraged me, because I had a hand in designing or working with designing a statewide rape kit, and she says, Pat, do not let them take the vaginal washing out of there. I mean, I've never seen anyone want it so badly as Kelly, but.
I think I think they went away from that because, of course, over time it was shown that a nice dried stain is very stable with DNA, and of course there's storage issues with washings. You're going to have to freeze it, So that's the whole deal. I think a lot of states went away from something like that. But they still had it in Louisiana when I got there, and I was like, what's a vaginal washing, But it's they do some type of washing with saline. I can't remember if they do it after the vaginal swab.
They do it after the vaginal swab.
Yeah, they do a vaginal swab, and then they flush the area with saline and then take it out. And it usually get like anywhere from like 1 mil to 5 mils. I think it's a 15 mil conical, and sometimes we have a large, And that's nice because it's not because you're not swabbing those epithelial cells out of the victim. There's not near as much of the epithelial concentration. I mean, you still have those there, but it's not near the same as it is from a vaginal swab. So that's why I like those.
I can tell you kind of where they came from, why they started in Louisiana, because they're so much easier to do ABO typing on the supernatant. I used to have to do, you know, the inhibition testing, the ABO inhibition, instead of do the ABO elution, absorption elution, we used to do the absorption inhibition. I know some of y'all may not even know what I'm talking about, ABO typing, but that's why it was in there, because it's a very easy sample. The seminal plasma has the ABO types in them in a water-soluble form. So we used to collect those, or they collected those in saline, so we could take the vaginal washing, spin it down, take off the supernatant, and immediately go to ABO typing. And they just kept on, and that's why. But they're really nice samples for what Kelly said. I have a feeling that some of these samples that are collected with vaginal swabs, they tend to scrub the interior of the vaginal walls, which is scrubbing off a lot of epithelial cells. And this method doesn't do that. This method kind of collects loose material and any material that's soluble in there. And you probably can sometimes get a lot better results, especially on differentials with the vaginal washings. Wouldn't you say, Kelly?
Yeah, I think since I've been working there, it does seem like I get a nice results with the vaginal washings. It's a lot, right? There's not so much female there, so it's just easier usually.
Just to clarify, when you were asking your question about how dilute of a smear, I have a mic.
Is it on?
It should be on. My cell pellet I do, traditionally when I was working in a lab, it was about 50 or lower the pellet. Now, I do 100 milliliter pellet, and I take microliter, sorry.
100 milliliter pellet.
It's late in the day.
That's bucket size chemistry.
Yeah, 100 microliter pellet, and I take three microliter spots. It's a 7 mil. Hers is 25 microliters from. And what's your original volume? Your pellet.
I've been doing about 75 micro. Like I'll leave about 75 in there, and my thinking is if I need to, I'm taking 25 off, and then I kind of leave that extra, pellets stored. For now, that's kind of what I'm doing, just in case I need to make another slide or whatever. But yeah, it could be like.
We do over the whole slide?
No.
We do a cellular
Okay.
extraction in about 300 to 500 microliters, and then that's compressed down to the
Right.
25 or whatever she said, 50, 75 microliters. So
But
we start with about 500 on
the actual
one swab.
The surface area that you spotted on. Do you know what that surface area
No.
is?
See, it's the pen slide. Well, you probably didn't notice because you guys didn't do them. But when you put the spot on there, believe it or not, the pen slide is sort of conducive. It spreads out. Because at first I was trying to kind of smear, like I'd put the spot and then I'd kind of smear it with the tip. But then I was having a hard time finding my cells because they were so spread out. So then I just kind of said, well, let me just put a spot on there. And they spread out a little bit. But I think the point Christine's probably going to make or was thinking of is maybe what's happening because I'm putting such a large amount on there, it's still not enough. It's not spread out enough. So maybe that's why I'm having a harder time. Whereas a smaller.
That's what I was trying. I was trying to gauge the comparison between how spread out yours was and dilute versus mine.
Versus a small, small spot. So yeah, there's definitely those issues. I suppose you could say, well, if you can actually see your pellet in the tube, chances are you have a lot of material there. So maybe you need to make sure you leave enough like PBS in there to kind of get it, more dilute. But yeah, that's definitely going to be something we have to consider.
More from a business logistics kind of standpoint, but looking at your process map that you were showing, and we talked to Kelly a little bit at lunch about it, I understand you're planning to use that process map for all of your samples, which would work well with a laboratory that has very few analysts.
Mm-hmm.
But I'm interested in like a laboratory like you guys with 100 analysts or whatever. How would you process map something like this through there? How many laser dissection microscopes would you need?
Well, how many 3130s do you have? One for each 3130. The same price. No, I'm really kind of
Actually,
joking
no,
with you a little
same
bit there.
price,
Huh?
Three.
We got three.
We have different kinds. We don't have them all. I mean, I don't know what we're gonna do, but I think we would need at least three because usually there's about 3 analysts who do rape kits each day at the OCME, and it's been pretty. The one thing that stayed the same is the number of rapes we've had. So there's about 3. we took more. So it hasn't. Our rapes haven't changed. We reprocessed them since Kelly left. all we've done is take different case types, like burglaries. And there's usually 3 or 4 people on sexual assault kits, so I'd say you would need 3, unless you would take turns, which you might be able to kind of do, but you probably would need more than 1, you need at least 2.
I mean, there's still the initial processing of the kit itself that doesn't require the microscope. You still got to make the slide. You still got to get your sample, so you could have a day or a half a day on the microscope looking at, say you process 5 cases the day before. You go in there and you get however time you need to look at 5 slides the next day. You've got your cutouts on there, and you go to the next step and do other processing. Well, you may have 2 carryover slides. Another person can look in the afternoon at that.
Well,
So.
having experience, we're working in that lab, which, and I think I was telling some of the people, the way it kind of works there, and I think it still does from what you're saying, is you have people on 1 week, all they're doing is they're looking at the sexual assault kits. So all week for 1 whole business week, every day they're taking a kit out. They're doing the screening, right? I forget, because you guys still do P30 ELISA.
Right, but I think what you were saying is you make the pellet,
Right.
you screen the slide, and then if you want to, you go back and take the sera, right?
No,
And do P30?
you can do the supernatant can still be screened ahead
Right
of the
then,
slide.
yeah.
You may prepare the slide, but you still have the supernatant to do your screening on.
But the nice thing is with them is what she's saying is, so then you'd have one person the whole week preparing, basically they'd be preparing a bunch of slides for LMD, let's say.
If you prepared the slide, you would do the P30 first as opposed to screen the slide?
I would probably go ahead and prepare the slide, but I would still do the P30 AP first, because you can do it on the supernatant. When you do your cellular extraction, like I said, we have 300 to 500 microliters of solution. Spin that down into the pellet. Well, right now what we do is we pull the supernatant off and drop it into an AP, a ceramic plate on the way, and then save the rest of the supernatant, and we do the AP test on it right there before we even looked at the slide.
Right.
So-
Then if your P30 was negative, you'd still look at the slide.
Yeah, you
Most
And if
likely,
it was
yes.
positive, you'd still look at the slide to dissect off the
Well, if
sperm.
it was positive, you wouldn't have to look at it too. You would look at it and cut at the same time.
Right, but in either way, you're still looking at the slide, so
Yeah.
do you still need P30?
Well, that's a whole nother ball of wax,
Well,
of course.
no, you wouldn't. Let's say you do it. I think it's the way we do it too. You do the AP test, you save the supernatant, and you make the slide. And then you look at the slide before you start into the process. Right now, that's what we do. You've made the slide, and you've started your cellular extraction. I know some, but you could examine the slide then. If you saw sperm, there'd be no reason to do the P30 after that.
Well, just for our lab, at least the way Pat taught me, to save cost, we try, because we have the ABA card, which is, that quick little, thing to tell you maybe you have male semen. So to save cost, I, of course, do the AP screen real quick, and then I look for the sperm on the slide. Because, of course, if you see sperm, that's a confirmation, and you can go. If you don't see sperm, then you might at least, like you said, take the supernatant. And, of course, like I said, we do the ABA card. Yours is different with the P30 ELISA. That's a whole different thing. Right, because you guys send the cutting off. I'm trying to remember how that went.
Yeah, like we take the anal, oral, and vaginal swab and the dried secretion swabs, and I could see making a slide for all of those and skipping P30. Right now, we send them to P30.
And you could do that.
And then maybe just go with that, and then I guess if it's negative, maybe do P30 to
Right.
make sure you didn't miss anything.
Mhm.
But then the
There'd
clothing.
be also information on what you want to do with that slide. Then you say, hey, I've got P30 on the slide. I don't see sperm. I do the sperm paint. Still don't see sperm, but you got P30. So now maybe that's not sperm in there. That is semen from a vasectomized individual. So now that says, okay, fish, because I want to see if I can find the male epithelial cells or male white blood cells. You see, that goes back to, here's the information I'm gathering on which step to take. Again, negative, say you had a positive AP, but you found no spermatozoa. And you say, okay, well, I want to test for P30. Okay, I'm going to test for P30. And then you say, okay, I got P30, AP, but I didn't see sperm under bright field. Okay, let's go to sperm paint. You still don't see sperm. But you still got the AP and the P30. So looks like there's semen there. So now the next step is, okay, I probably maybe have azospermic male or vasectomized male, and there's a chance I'm going to have white blood cells in there or other epithelial cells. So now that would be a good indication to take that next step to go to the chromosome paint and see if you can find any male cells in there. And then if you don't find any male cells, you say, well, if there's some reason that's in there, just cut out the deal and do an extraction and try Y-filer. So Did you follow my process?
Yeah, I mean, we skip the chromosome paint because you can't buy it now.
What I'm saying is, but if you could, that's
If you
what
could, yeah.
that's the process where you could go through it, the whole thing, and test it.
I think the whole thing, though, the whole process will take a lot longer than it currently does because
Yes.
of all of our
Well-
batching. But if one thinks that we could be missing, that what we're calling negative may not necessarily be negative because there could be a few sperm left,
Right.
is that worth the extra time for all the other cases?
Right.
And I think for it to be accepted in the industry period, we have to have some metric that measures that. I mean, I don't think any laboratory system is gonna buy something that's gonna create backlog.
Yeah.
We just have to throw more people at the sexual assault team.
And throw more money at the people.
You know, it comes to the point. Are you going to leave it laying on? Are you going to leave the evidence laying on the table? And right now we have to. I mean, there's no question that we can't work every one of the cases that come through to the best extent that we possible. So here's the quality issue: can I work all the cases to the best I can, or do I have a certain sacrifice a bit of quality or numbers quality to get higher throughput on the cases we can, and realizing that maybe what's left up here that we can't examine, we really don't have the tools to do it anyway.
Well, and I think too, because it does seem like obviously if you were to implement this whole process, doing chromosome paint and everything, I mean, that clearly almost makes more work for us because now we do have the ability to separate the male and female, let's say, skin cells. I mean, obviously this is, now it might make more work, but, you know, it could be one of those things where you implement it for special cases, like a child, you know, molestation, where, yeah, a lot of times you just have, you know, just, you know, digital penetration or whatever, where they're, you know, a child, they can't speak.
Oral genital contact
Yeah,
is
or,
common
you know,
for child abuse.
But I could see like a larger lab, you know, the person's doing the sexual assault kit screenings, doing their screening and making a slide, and so now they have a bunch of slides, and then if it were P30 positive, they could go on, or they could cut the spot out, you know, whatever. But
I mean, that's a good point on the child abuse cases where you have oral genital contact, where you're negative AP, and you're going to be negative AP, negative P30, and negative sperm, but you have very good indication that there may be epithelial cells there from the person that did the abuse. Well, if you don't have the fish capabilities, there's nothing you can do with it except for do an extraction and hope for the best. But in this case, at least you would, with the fish, you would have the capability of taking that swab and finding perhaps the male cells on the young female's genitalia. And you say, hey, here is a nice straight profile. Right now, we might be able to do the swab and do the extraction, and with luck, get a major contributor or perhaps a clean sample. But this would be one way which, if it's there, you could definitely find the male cells. And then again, if you can't find the male cells, you just simply have to cut out the material and do your standard extraction. So it gives you capability, a new capability that you don't have now. Yeah, it's going to take a little longer time, but you know, when back in the days when DNA analysis was just DQ alpha, that was really short. Now we've got the STRs, the mini STRs, the low copy numbers. Have we not increased our workload in other ways just to do better analyses?
that was the point I was going to make, actually, even like just with the little copy number. I mean, I don't know about, you know, all your places and what your policies are, but now at our lab, of course, I mean, they want DNA testing done on everything because just touching or whatever, low copy number is opened a whole nother batch of cases. I mean, so yeah, I think it's sort of relative. Again, you know, it's kind of relative.
You actually made an interesting point earlier, and that was that you said that you can't separate multiple male contributor mixtures. The same would apply with the staining for male and female epithelial cells.
Yes.
Right.
So at what point do you establish I've collected enough cells to ensure that I am seeing the true profile that's there? You know, if you're collecting 5 cells.
Yeah, I think, did I talk to you about that the other day? If you go and you collect 5 spermatozoa, and it's from 2 different males, so you have 2 from one, 3 from the other, so you get this mixed profile where it maybe really doesn't look like a mixture, looks like a single source profile because you have 2 alleles at every locus, but it's actually a combination of 2 males with a lot of dropout.
For 2 haplotypes.
Now, or 2 haplotypes. Well, yeah, 2 haplotypes. So now you got this profile. It looks like a single source profile. It actually excludes both of the individuals.
But if you did it in replicates, you would see that.
Yeah.
Hopefully.
That's right. We did it.
Perhaps not.
No, you would. If you did it 3 times,
Yeah,
you would see it.
But
3 times with 5 cells each? Well, maybe
You
so.
would. You would
But if
say-
you had,
you would call it a call that a possible mixture if we're not 100% sure, but we err on the
Yeah.
conservative side and consider
What would
it
you
a
use for your
mixture.
composite profile?
But what
I
if you
would
just,
treat it as a mixture and look at all three amplifications concurrently.
well, hopefully on a microscope, what if you were able to cut all 15 into one tube?
We would divide that and amp it 3 times. That's how we'd process it. I wouldn't want to risk making an error from one amplification.